Hi everyone, welcome to Fleshing Out the Facts on Duodenoscopes. My name is Mindy Benedict. I am the current Global Infection Prevention Manager here at Olympus, and I'll be your hostess today. Today we'll be sharing some updates we've made to our reprocessing tools and manuals based on the learnings we have taken away from our human factors study. We'll also go through duodenoscope reprocessing and discuss the safety and efficacy with one of our experts. And finally, we'll go through the new TJF Q190V duodenoscope design, including the disposable distal cover and manual reprocessing adapter. First, I'd like to give you a high-level overview of what a human factors study is. The main objective when performing a human factors study is to determine if reprocessing personnel can understand and follow the manufacturer's recommended reprocessing instructions as written in our IFUs or instructions for use. The general study design includes a simulation environment where trained healthcare facility staff will reprocess the device in accordance with the reprocessing manual. These trained healthcare facility staff are the reprocessing technicians and nurses who would normally be doing this job on a daily basis. During a human factors study, human factors moderators record each participant device interaction. They watch as each participant reprocesses the device according to the manufacturer's instructions. Deviations from the instructions are recorded, and the moderators will then define any deviations as critical failures, use errors, close calls or near misses, and reprocessing difficulties or hesitations. Reprocessing difficulties or hesitations are those steps where the participant may have had a slight confusion or a hesitation, but did eventually perform that step correctly. Then, the moderators will hold an interview with each participant. Well, they ask them critical task questions where they analyze the participant, the IFU, such as asking them if they know why they're doing a certain step or what a certain step achieves in the process. Now we'll review two high-level findings from our Olympus duodenoscope human factors study. The first major finding was that we found that some technicians had difficulty with any multi-step tasks that are involved in reprocessing duodenoscopes. An example of a multi-step task is during pre-cleaning when the technician needs to be aspirating water or detergent while at the same time raising and lowering the forceps elevator for a defined number of times. The second major finding was that we found success varied by participant. Almost half of the observed pre-cleaning errors were attributed to only 3 of the 15 study participants, and half of the observed leak testing errors were attributed to only 2 of the 15 study participants. So now that we've gone through what a human factors study is and what some of our high-level learnings were, we're now going to talk to Eric Smith, one of our Olympus senior technical trainers, on the modifications that we've made to our reprocessing resources and manuals.
Thanks, Mindy. You know, I really love when an author utilizes foreshadowing to really bring out the greatness of a good book. Here's my attempt at foreshadowing. I'm going to share a little quote that I read the other day that I think really leads us into what we're about to talk about. Change means that what was before wasn't perfect. People want things to be better. That's what I feel like we as a company are doing here at Olympus. First, I will start with reviewing the changes we have made to our current reprocessing resources. From our findings to the human factors testing done with the TJF Q190V, we identified a number of common use errors. To draw further attention to these steps, we modified the TJF Q190V reprocessing videos to include a small red rectangular caution call-out on the common use errors. Errors commonly missed or done incorrectly. This will bring attention to the viewer and refresh on the proper way to complete these steps. Olympus has also put together a human factors landing page on our medical website with information on findings and resources for facilities to utilize.
That's great, Eric. Many of our customers know about our on-site reprocessing training, but could you tell them more about what we have recently developed for digital learning?
Absolutely. This is definitely something to get excited about. Olympus has taken training on the reprocessing of our endoscopes to a whole new level. We have partnered with Amplifier to create a digital learning module on the reprocessing steps of the TJF Q190V. This learning module uses learning algorithms, knowledge analytics, and diagnostic capabilities to drive improved outcomes in duodenoscope reprocessing. The module can be found on the landing page shown on this slide and is completely free for participants. It is an interactive experience that includes knowledge checks for continued learning. The results can even be shared with the participant's manager for further teaching and assurance of understanding. We're putting together a similar module for the TJF Q190V that is expected to launch soon. Mindy, you can see some of the modifications we have made to current resources, but we've always been making modifications to the TJF Q190V resources as well. Let me just start by saying that when you have a product, you have to have an end goal that you must strive for. Otherwise, the product cannot be a success or make it to the market. The end goal for the TJF Q190V was ultimately patient safety, and I truly believe we have met that goal. Remember when you were a kid and used to ride in the very back of your parents' station wagon, jumping around, playing around, or even maybe drink directly from the hose? Well, this isn't your father's reprocessing manual. Times have changed. We are more aware and concerned with eliminating gray areas. What we did before writing this reprocessing manual was took what we learned from the human factors study performed for our other ERCP scopes. We took feedback from you, the end users, and we made the manual easier to read, understand, and execute. The steps are more clearly written out, so it may appear as more steps, and it may seem like it is a lot more pages, which is actually true, but that's due to the amazing pictures that have been added to help guide the user through the process. And you know what, Mindy? That's not all. We are currently in the final stages of updating this reprocessing manual to make it even more better. Yes, more better. It will be in color. Even more pictures, better chapter headings to make it easier to navigate. Just all around more better.
These IFU changes are exciting, Eric. Thanks for sharing this with us. We're excited to share these tools and partner with our customers on reprocessing training to continue these learnings to improve duodenoscope reprocessing. Before moving into reprocessing efficacy and safety, I'd like to touch on current reprocessing guidelines for flexible endoscopes. I'd like to point out that high-level disinfection is still recognized as an acceptable standard for reprocessing semi-critical gastrointestinal endoscopes by many professional societies. These societies include the SGNA, ASGE, ACG, AGA, and more. Some guidelines may begin to recommend moving to sterilization for semi-critical endoscopes, but this is not a requirement. It is only a recommendation. As we talk about reprocessing training and resources, I want to move into the efficacy and safety of reprocessing duodenoscopes. This includes the efficacy of the solutions used to reprocess duodenoscopes and the impact these solutions may have on the environment. For this, I want to bring in Jeff Daniels, our Director of Marketing for our infection prevention products.
Thanks, Mindy. I would be happy to go through reprocessing solutions at a high level. I think it's important to set the ground rules here and the foundation. Any reusable device needs to be reprocessed before being used on another patient. And reprocessing is that process of rendering a reusable medical device safe for that next patient. This includes cleaning, testing, disinfection, among other steps. And as part of the disinfection steps specifically, disinfectants are used to clean that device and to kill the microbes that are on it. Disinfectants are regularly used for reprocessing these medical devices, and they rely on the use of appropriate liquid chemical germicide as that step. Efficacy is proven and cleared through the Food and Drug Administration, and it provides suggestions on testing and proof for the 510K clearance of that disinfectant and that LCG for use. So I've mentioned liquid chemical germicides a few times and how medical devices are regularly used liquid chemical germicides to be disinfected. There are multiple products that are out there. You may see Parasitic Acid, you may see glutaraldehyde. Orthothialdehyde is commonly called OPA, is regularly used, as well as hydrogen peroxide and hypochlorites. These all have their pluses and minuses, their pros and cons. Some are useful in manual disinfection, others are for automated reprocessing only, and only suitable for AERs and use in AERs. Each has its benefits and drawbacks. Parasitic acid specifically has become a widely used disinfectant used in flexible endoscope reprocessing specifically, and is used in several automated reprocessors. It's the primary LCG used in the Olympus automated reprocessors, and we use the brand name Aesocide C for our parasitic acid in the OER. I've mentioned Aesocide C as a parasitic acid, and parasitic acid is made from a combination of acetic acid with hydrogen peroxide. This makes the parasitic acid more effective than acetic acid alone, and it also gives it that distinctive vinegary smell that comes from the acetic acid aspect. It's formed through that combination. When you combine those two, you get parasitic acid and water as the byproducts. And key to that is parasitic acid as a liquid chemical germicide. It's microbicidal, it has no toxic residue, and it's not bioaccumulative when used as a disinfectant. So I mentioned that parasitic acid is highly microbicidal, which is very important when reprocessing a medical device. Our goal is to kill the microbes that reusable device was exposed to, and to prevent any contamination from remaining on the device. Parasitic acid has been shown to be effective in multiple tests. Here are a small sample of the results, and of course these are for Aesocide C. Other manufacturers may have their own formulation. Key to important to know is that it's effective against a wide range of microbes. It's bactericidal, kills bacteria. It's tuberculocidal, kills Microbacterium, terrae, things like that. It's fungicidal, it kills different types of funguses. And it's virucidal, it kills a wide variety of viruses as well. It does not build resistance. There are no reports of parasitic acid resistant bacteria, and it does not fix protein soils. In fact, it eases their removal. You can see from these charts some of the relevant kill times and how long exposure it takes to achieve that for both bacteria and viruses. You can see efficacy against a variety of other test organisms as well. The contact time for liquid chemical germicides is significantly increased over these contact times to ensure a wide safety margin. I do need to remind you, this is based on thorough and meticulous cleaning before the disinfection process. So again, we've mentioned that Aesocide C is Olympus's parasitic acid product, and it's been used in the US for almost 10 years now. It's been even longer in other parts of the world. The Food and Drug Administration has cleared it for use in the OER, and they've picked that for a variety of reasons, or cleared it for a variety of reasons. It is environmentally friendly with its byproducts, which are basically acetic acid, oxygen, and water. It's fully automated in the OER. It has a standardized cycle, so it's very consistent in its use. It may enhance the removal of organic material and endotoxins. It has no adverse health effects to operators under normal operating conditions. It's compatible with many materials and instruments. It does not coagulate blood or fix tissues to surfaces when used. And it's a sterilant that flows through scopes, facilitating salt, protein, and microbe removals. Drawbacks do exist, so nothing's perfect, but it includes that it may not be materially compatible with all products, and it is not suitable for manual reprocessing. It should only be used in an automated reprocessor. So I stated earlier that parasitic acid is formed from acetic acid and hydrogen peroxide, and it makes it more effective in this state as a disinfectant. It also begins to break down after use, returning to the component materials such as acetic acid, oxygen, and water, and leaves it with no toxic residues and limited byproducts, the buffers and such that are included in Aesocide C. As a result, there are no long-term toxic effects. There's no teratogenicity, and there's been no sensitization recorded with the use of parasitic acid. So because it's an environmentally friendly disinfectant, parasitic acid has begun to be used in other industries and with other experiences. Parasitic acid is increasingly supported in the use of the food service industry, where it's used for surface sanitation and packaging processes. It's also widely used in wastewater treatment, where previous disinfectants could actually harm aquatic life when flowed downstream or from the chlorides that are included in there. So in total, I think we can agree that parasitic acid is generally considered to be a low-risk disinfectant. It's effective against a wide range of microbes. It breaks down to acetic acid, oxygen, and water with no toxic residue. There are no long-term toxic effects that have been recorded, and it's considered to be environmentally friendly. Olympus is pleased to offer Aesocide C as our parasitic acid in consideration for use in reprocessing your duodenoscope.
Thanks for that deep dive into reprocessing chemicals, Jeff. Before moving into the new Olympus duodenoscope design, I'd like to give you an overview of the FDA recommendation on duodenoscopes. In August of 2019, following the 522 and human factors studies, the FDA made a recommendation for facilities to begin to transition away from duodenoscopes with fixed end caps to duodenoscopes with innovative designs that can be reprocessed more effectively. This recommendation includes duodenoscopes with disposable components, such as removable end caps, and disposable scopes. Olympus has since launched the TJF Q190V, our newest model of duodenoscope that includes a disposable distal cover and adheres to these FDA recommendations. Olympus will be conducting a 522 post-market surveillance study of this scope beginning in the winter of 2020. Now we'll be talking to Amanda Call, BSN, RN, CGRN. Amanda is one of our clinical specialists here at Olympus, and she'll be telling us about the TJF Q190V design improvements.
The major design change made to the TJF Q190V is the addition of the disposable distal cover. In August of 2019, the FDA made a recommendation for facilities to transition to duodenoscopes that have disposable components based on the 522 study findings, and the TJF Q190V meets that recommendation. As you can see in the image below, the TJF Q190V distal end does not have a fixed end cap, but rather requires the MAJ 2315, the disposable distal cover, starting at a list price of around $5 per cover. Having this feature allows the technician to have easier access to the distal end when flushing and brushing, and better visualization of debris. Personally, in the field, I have witnessed this with facilities that are transitioning into this new TJF Q190V in that they have absolutely had better visualization of the distal tip once they have removed the cap. We have also received images and videos from accounts who have removed the cover and found stones and sludge wedged behind the elevator that they would not have been able to see in previous generation scopes. Coming from a very busy ERCP procedure world prior to joining Olympus, I know firsthand what it's like to pull a scope out of a procedure room that is covered in sludge and debris, especially in those fixed end caps from my experience, to now how much easier it would be to be able to visualize behind the elevator and to remove that stone and sludge. Many times for me, that meant passing on to the tech in the decontamination room the amount of stone and sludge in the case to remind them that it may require some extra brushing during their manual cleaning phase. Or worst case scenario, I was the one reprocessing that scope on the weekend, and I knew just the extra work it would require of me during the manual cleaning phase to visually inspect and make sure that all the stone and sludge and debris were removed. I have a video to show you on the attachment and detachment of the distal cap. It is super easy. You simply line up the open side of the distal cover with the lens side of the distal end of the duodenoscope, and you push down until you feel the opening of the distal cover go over the hook of the distal ring on the scope. Then to remove, you tear along the perforated line along the top of the distal cover and twist the distal cover until it is removed from the scope. As a nurse in the busy endoscopy lab, we definitely like accessories that become easy to use and are not difficult. Many of our staff rotate through the procedure rooms and are not consistently in one room week in and week out. As well as some of these procedures happen outside of our endoscopy lab, right? Sometimes we have to travel to the OR or to the fluoroscopy unit, and we want something that is easy for all the staff to use, that's very consistent across the board. It's 2020, change is inevitable in the medical field, and we like things that change for good to make the life of the nurse and the tech significantly better and help to make our time be more beneficial with our patient. The last feature of the TJF Q190V that I want to show you, without stealing any of Eric's thunder on reprocessing, is the MAJ 2319, the manual flushing adapter. Because of the TJF Q190V features the disposable distal cover, this allows the reprocessing technicians and nurses to have access to the distal end while reprocessing, as we can clearly see. With this access, Olympus has developed a flushing adapter that allows for consistent, repeatable flushing onto the directed areas on the distal end of the duodenoscope. I will play this video to give you an idea of how the adapter works. As you can see, there are 4 holes in the adapter that flush the targeted areas of the distal end of the scope. You can see these areas in the images to the right of the video. It is simple. You place the distal end lens side down into the green side of the adapter, snap to close, and follow the reprocessing instructions in the IFU. In the endoscopy lab, the nurses and technicians like things to be simple, because that helps it to be consistent for all staff. Our goal as the endoscopy staff is to take care of the patient on the table, as well as the capital investments and hospital equipment that we are provided to work with day in and day out.
Thanks, Amanda. So in conclusion, we've looked at how reprocessing tool modifications have been made to reflect human factors learnings, that improvements to current and future reprocessing IFUs have been and will continue to be made for increased understanding of reprocessing steps. We see how duodenoscope reprocessing is safe and efficacious. And we also reviewed TJF Q190V design improvements and how they allow for easier access to the distal end, and this includes a specialized reprocessing adapter. Thank you everybody for joining us today.